In prokaryotic cells, translation happens simultaneously with transcription. For this example, we'll be talking about translation in eukaryotic cells. Starting off, mRNA will leave the nucleus through nuclear pores and then into the cytoplasm.
Pause.
A ribosome will then attach to the mRNA, and the ribosome is also responsible for translating the nucleotide sequence of an mRNA molecule into the correct amino acid sequence for a polypeptide. This is done through reading codons on the mRNA, a 3-nucleotide sequence that codes for specific amino acids based on the order and types of nucleotides. tRNA has a nucleotide triplet to match the mRNA, known as a codon, an anticodon, which allows specific and proper amino acids to be brought into the polypeptide chain later on.
Alright, pause.
An enzyme called aminoacyl tRNA will attach the amino acid to the tRNA, leaving us with a tRNA ready for protein synthesis. Moving on to the ribosomes. Ribosomes are made of about 1/3 proteins and 2/3 RNA. The ribosomes also have many other components, having a large subunit containing an A, a P, and an E site, with an exit tunnel, a small subunit, and an mRNA binding site. The aminoacetyl tRNA binding site, or A site, holds that tRNA in place while waiting for the amino acid to be added to the polypeptide chain. The peptidyl tRNA binding site, or P site, holds the tRNA that's attached to the growing polypeptide chain that will leave through the exit tunnel.
They want to interrupt because you guys are talking, so I thought I'd
Finally,
do
we have the exit site, or E site, where the tRNA leaves the ribosome. Starting off, the small ribosomal subunit binds to the mRNA that travels from 5' to 3'. An initiator, tRNA, will then bond to the start codon, causing the large ribosomal subunit to attach, completing the translation initiation complex. The complex does its job
Is that how you greet each other?
Pretty much.
Wow, that's like an old couple, right?
Yeah.
You've been going out too long, I guess, huh?
Not
Kidding.
even, hasn't even been that long.
Oh, and you, okay.
properly recognizing codons, forming peptide bonds in the polypeptide chain, and sending tRNA to get amino acid. And then here I'll just I won't have any audio, and it'll just be you doing it.
Yeah. You know what? You could time lapse this a bit.
Yeah.
Bye, have a good weekend. You have a good weekend. See you later.
Basically, you're just gonna move. Basically, you're gonna move this, and you're gonna tap it here, put that together. Polyribosomes, or chain multiple ribosomes, are able to read the same strands of mRNA to produce multiple of the same protein at the same time.